Hello, we have a fantastic presentation in store for you, exploring how alternative splicing and gene isoforms influence disease, featuring our esteemed speakers, Dr. Megan Noonan and Dr. Christophe Fleury. Megan and Christophe will be sharing their expertise on alternative splicing in kidney disease, which we're eager to learn more about. Before we dive into the presentation, let me introduce Megan and Christophe. Megan Noonan is a postdoctoral researcher at Washington University School of Medicine in St. Louis. She completed her PhD in medical and molecular genetics at Indiana University. Her research aims to uncover novel mechanisms of kidney disease progression via dysregulated alternative splicing. She uses nanopore sequencing to better resolve isoforms for more accurate alternative splicing analysis in bulk and single-cell RNA sequencing datasets. Christophe holds a PhD from the University of Caen in France and has subsequently worked for several years in academia on a range of topics in the field of host-pathogen interaction and gene regulation. He made the transition to industry over 10 years ago, holding technical and commercial roles in companies working at the forefront of genomics. These experiences have provided him a vast expertise in genomics and its applications, including transcriptomics, single-cell sequencing, and spatial transcriptomics. Now, without any further ado, I'm delighted to hand over to Megan and Christophe to share their insights and knowledge with us.
Good afternoon, and thank you to Nanopore for allowing me to come and talk about my research. So the aim of our group is really to understand the molecular mechanisms that drive kidney disease, and in particular, I'm interested in what drives diabetic kidney disease, for the purposes that diabetes is the leading cause of end-stage renal disease worldwide. And currently, the therapies that are used to treat the progression of this are lacking. And so there's a need to discover novel mechanisms to create new therapeutics to create a better um, thing for these patients. And so through a large body of work from our group and others, which I've referenced down here, using single-cell RNA sequencing short read, they found that there is a decrease in this factor called SRSF7 in the proximal tubule cells of kidneys. And these are serine arginine-rich splicing factors, which are shown here. It's a family of 12 different splicing factors. We're focused on SRSF7, and these are involved in alternative splicing. And basically, they aid in the recruitment and assembly of the spliceosome, which is the machinery necessary to perform splicing, where you splice out introns or exons. And you can see this tiny orange factor, which is SRSF2 in this picture, but SRSF7 performs in a similar manner at the beginning of this entire process to recruit these factors. And the point of alternative splicing is really to create transcriptional diversity. From one gene product, we can get many different isoforms that might have different functions. And so these are the main alternative splicing patterns that have been observed. They're not exhaustive, but this can lead to altered function due to splicing in or out of various functional domains, as well as altered expression due to either a non-coding transcript or something that is due to nonsense-mediated decay. So alternative splicing is also this additional regulatory mechanism outside of just expressing a gene. So there are various tools to assess RNA splicing. In the past, it was mainly using short-read RNA sequencing. However, as many of you probably know, this is lacking as far as how it can resolve individual isoforms because they have to be inferred. And so with the invent of long-read, we are now better able to resolve individual isoforms, usually the entire thing. And so this allows for more accurate transcript discovery and quantification. So to understand the function of this factor in kidney cells, I just want to give you guys a brief reminder about the kidney. It has an outer cortex and an inner medulla. And within the kidney, the functional unit is called the nephron. And we tend to focus on the proximal tubule cells, or the PCT cells, that are in the cortex. These are the most prone to injury and also highly metabolically active. And so we utilize this cell type. There is a immortalized human cell line of renal proximal tubule epithelial cells that we use to test this. And so we treated these cells with siRNA against SRSF7. and then submitted the samples for both short and long read sequencing. And I'll mainly focus on the long read results. And I just want to show an example of like what comes off the sequencer, which is just, this is showing the distribution of read lengths from this experiment up to like 4 kb. So seeing that we do indeed get long reads from this. And then when we look at the, after aligning and putting it into IGV, we can compare and see the differences between short and long reads. So at the top are the short reads, and they're just little snippets trying to figure out where they are. And then with our long reads, you can see this is the SRSF7 gene, which runs right to left. And many of the reads are, 1,800 to 3,500 base pairs, and they are covering the entire gene. So we're getting a better picture of what is actually happening at the isoform level. I also did an analysis to measure exon-exon junctions between the long and short reads for each sample. This was done using reg tools from this reference, and we found in every sample that long reads, even given the difference in coverage, were able to detect more exon-exon junctions, which is very critical for alternative splicing analysis because that's what is typically used to understand the splicing that could be occurring. To understand the function of this, after we performed our RNA sequencing, we did gene set enrichment analysis and showed that in our in our knockdown cells, that they had enrichment for inflammatory pathways, and then also noticed in our normal cells they were enriched for like cell cycle pathways, which had us hypothesized that there might be some proliferation effect on these cells. So we did measure MKI67 expression and saw that this was indeed markedly reduced, and also did BRDU incorporation to look at the cell proliferation again and saw that this was also lower with SRSF7 knockdown. So not only does this factor, at least when it's present, seems to help preserve this, keep a anti-inflammatory environment as well as maintain proliferation, since when we don't have it, we have inflammation and a decrease in proliferation. Another analysis we did was using this package called Bamboo, which is specifically made for long reads and can quantify as well as do differential transcript usage analysis to understand the alternative splicing that is occurring. So we ran our RNA-seq data through this package, and it identified over 500 genes with differential transcript usage. And then we did gene ontology analysis to see which pathways these genes were involved in. And interestingly, again, we see these genes are involved in inflammation and cell cycle and also in splicing. So I just want to give one example of a gene we found that was in a cell cycle pathway called BANP. And interestingly, when we did this same analysis in our short read, we didn't find this gene. So this was only found with our long read sequencing data. And we use this package called isoform switch analyzer, which helps predict isoform switches as well as any potential functional consequences of this. This is what the colors indicate. And so from just this one gene and this analysis, it predicted an increase in this 204 transcript and a decrease in the 228 transcript. Another thing I really want to point out is the gene expression between the two conditions is not different. So this is something you would miss if you just did gene level analysis. And then to validate this finding, I designed primers against basically the difference between the two transcripts was the inclusion or exclusion of this exon 5. So I designed primers to flank that exon and performed RT-PCR. And as you can see, we do in fact have a decrease in the 228 transcript and an increase in 204. And so as you can imagine, one way we can also test the function of this, since this is a cell cycle gene, is to target this individual transcript and see if it would have effects on cell proliferation. And this is something we want to do with not just this gene, but many others for future studies. The next question we wanted to know was, how does splicing change across mouse kidney epithelial cell types? And the perfect way to assess this is by using single cell long read sequencing. And so we use the 10x Genomics platform to do our single cell, and then Nanopore has a part of this workflow. So we start with our cells. So for this experiment, I used 3 different mouse kidneys and did single cell dissociation. We used the 3' GenX kit and targeted 10,000 cells. I will say that is a lot. and it does affect the coverage of your genes or transcripts per cell. So just keep that in mind if you're looking into your own experiments. And then you kind of follow the same 10x workflow until you get to the full-length cDNA generation. And because we can now sequence full-length cDNA, we just submit this for the sequencing or the library prep and then sequencing. And for this experiment, we did one sample per flow cell. Again, this is probably not ideal with the amount of cells we targeted, but you can always sequence across more flow cells. And then finally, we used the Epitome Labs workflow for single cell to analyze this data. So I just want to start with some read and workflow statistics. This is just highlighting things like the median read length, which was over 800 for each sample, which is pretty typical for single cell RNA-seq. And then our median read quality was 22, which was much higher with this new implementation of Dorado. I had done some previously where it was much lower. So this is just continuing to improve. And then finally, from ONT's workflow, it will tell you how many high quality barcodes it has identified or number of cells. And we can see for each sample it is around 10,000. So that's kind of on target. And then we get the expected number of genes and transcripts. And again, you'll notice median reads per cell is pretty low, but it is enough for us to detect the cell types that we expect, which I'll get into. So from this data set after QC, we identified over 16,000 cells from these 3 mouse kidneys across 20 different clusters. We then used our sort of in-house and published cell type annotations to annotate these cells. And we come up with this single cell UMAP showing very typical cell types that we would expect in the kidney, even some that are harder to detect with single cell, including podocytes and certain immune cells like T cells and B cells. So we were really pleased to see that, even given the lower coverage. Another thing we had noticed is that our very canonical cell type markers were consistently kind of split across 2 different cell clusters. And I'm not entirely sure why this is happening, but we are wondering if it is due to the long reads that we're getting now with this single cell workflow. And just to kind of show you what I mean, SLC5A2 is a very canonical PCT marker, and you can see it pops up not only in one, but another cluster, and same with SLC7A13 and SLC12A1. So as of right now, I'm treating them as different. I'm not sure it will stay that way, but it is quite interesting that we're seeing this with just our long read data. Our lab has done a ton of short read kidney stuff, and we've never seen this, so it's quite interesting. One thing I did do was ask, what is the difference between shared cell types that are split, and across the board for each one, they seem to have changes in alternative splicing processes. So this is really interesting for us, wasn't really expected, and even our gene SRSF7 pops up in here, so that's also really interesting considering these are just normal kidneys. So we also did transcript level clustering. Again, the advantage of doing this is not only are we getting gene per cell, but also the isoforms per cell. So I just took the cells we had already annotated and clustered based on the transcripts, and we identified 21 clusters and ended up projecting back the gene level annotations or cell types that we identified. And as you can see, each gene cluster is kind of spread across different transcripts and kind of vice versa. One transcript cluster actually identifies different gene cell types. And again, I haven't delved too much into this, but this is a really interesting area for us to kind of start to define the transcript level landscape of kidney. We also wanted to compare our long read data set to a published short read data set that was also using single cell. So we used this paper here. Again, it's a mouse kidney, and we just did this confusion matrix to look at consistency between what we called a cell type versus what they called it, and pretty decent consistency across annotated cell types. And I will say, with the new implementation of Dorado, even from that part of the analysis has greatly improved this based on, I don't have the other data set, but it looks a lot better. So I think everything seems to be improving for this kind of workflow. And then finally, the entire point of this is to look at alternative splicing between cell types. So for the sake of brevity, I'm just going to focus on looking between PCT1 and TAL1. And just to remind you guys, these have different spatial locations within the kidney, and they also have different functions. So to do this DTU analysis, we use this package called dturtle, which is specifically made for single cell analysis. It isn't necessarily for long reads, but we did use it for that. And this package identified almost 600 genes with differential transcript usage among over 1,100 transcripts. And these are the top 10 genes that, like most significant genes that were identified between the PCT and TAL. And so we're going to focus on Claudin 10. When we saw this, we were really interested because Claudin 10 is actually a very common TAL marker, and we see it come up for PCT as being significantly differently used. And so from this package, it gives you this kind of output, which kind of gives you the structure of the isoforms that are changing and tells you to what proportion. So in this case, the 201 transcript is used 90% more in the PCT versus the TAL, whereas the 203 is used 90% less in the PCT, or it's used more in the TAL. And so to kind of validate this finding, I designed probes for in situ hybridization and performed that in mouse kidney. We can see Claudin 10 didn't come out very well, but we would expect it to be in the cortex because that is what this is telling us, that it's in PCT cells. And then Claudin 10B we do see is very clearly in the medulla. So we're able to, especially when genes have different spatial locations, can validate this with in situ hybridization. And finally, this finding is not new. Actually, in 2009, it was published using different methods that the Claudin 10 isoforms are spatially located in different spots. So using RT-PCR, they had showed Claudin 10, again, is in the PCT, and Claudin 10B is in the medullary thick ascending limb. And when I look across our data set at the two different transcripts, we can very clearly see the separation of Claudin 10A in just the proximal tubule cells and Claudin 10B in the TAL cells. And the reason this is important is it leads to a functional consequence of the different expression of these isoforms, and that is when it comes to perm selectivity or preferring anions or cations. So in the case of Claudin 10A, it has this extracellular loop domain, which has a different sequence than Claudin 10B. And so in 10A, this loop sequence has a net charge of +5, so it will prefer anions. And then Claudin 10B has this net charge of -1, so it prefers cations. And because of this, the cells now have different functions along the nephron. And so just really highlighting the importance of doing this. And of course, we want to find novel isoform switches that could have functional consequences that we can test and validate in vivo or in vitro. So with that, I just like to summarize and lead into the future direction. So I've shown that loss of this factor SRSF7 in proximal tubule cells upregulated inflammatory pathways and decreased cell proliferation. More exon-exon junctions were detected with bulk nanopore sequencing samples, and this allowed us to discover differential transcript usage that isn't found with short read. Our single cell nanopore sequencing showed these split cell type clusters that we're still trying to understand and define. And finally, DTU occurs between the PT and TAL in mouse kidneys, and they do have functional consequences. And the future directions of this work is to investigate alternative splicing in mice that lack SRSF7 in the kidney, because this would help support our hypothesis that this will cause renal dysfunction, and supporting that changes in alternative splicing can drive kidney disease. And so with that, I'd just like to acknowledge the Humphreys Lab at WashU. Also, a special shout out to Jennifer at Nanopore, who helped me with the base calling, and then my funding sources. Thank you.
Thank you, Megan. Now I'm gonna be pleased to introduce you to nanopore sequencing and to show you how nanopore sequencing can make a difference in the study of the transcriptome. So first, a few numbers. 95% of human genes are multi-exons and undergo alternative splicing. 15% of human hereditary diseases and cancers are associated with alternative splicing, and 1 in 6 cancers are driven by gene fusions. And that is for what we know. You see here that we estimate that between 4 and 6 transcripts isoforms on average are generated per gene in the human genome. This number in particular is not fully precise because the reality is that the current technologies don't allow to measure precisely the isoforms and to quantify them either. Now, we live in a world full of genomics, and you might wonder why I say that current technologies cannot map efficiently isoforms. So first, we're going to start with one example. You take here this gene. This gene in this ideal scenario has only 4 transcripts that use different exons. With short reads, you're going to have results that look like this, where each exon is covered by multiple short reads. Short read sequencing will enable to quantify transcription at the gene level, but if you look at those different transcripts here, we might be able to detect the breakpoint of a fusion, and you might know that these fusions or isoforms are present, but it's going to be extremely difficult to quantify them. If we look at the extremities, those blue reads, we don't know to what transcript here they belong to. They might be to one or the other, so we can't quantify the isoforms. Similarly, we see here a breakpoint for one isoform, but if we were not sure that we have 4 isoforms, we wouldn't be able to determine if this one belongs to the 3rd isoform or to a 5th one that has been unraveled. With long-read nanopore sequencing, you see there that every read will span the entirety of the isoforms, and we can therefore detect them without any doubts, detect the novel isoforms, but also precisely quantify the isoforms. What I'm saying about the isoforms is also true for gene fusions, for instance. We will be able to quantify also allele-specific isoforms, determining whether what we're looking at come from the parent, from which parent it comes, the mother or the father. So how are we able to read through a whole transcript? The secret lies into the unique nanopore sequencing where we have a pore, and I'm not going to go into the details, we have a pore through a membrane, and each DNA or RNA fragment can go through and will be sequenced whether it's short or whether it's long. We have no bias regarding the length of the fragment that we can sequence. When it comes to transcriptome analysis, what are the options that are offered to you? You can look directly at the RNA because our technology enables to sequence directly RNA with different flow cells. And this approach is a fantastic research tool that will allow you to look at modified bases and see their role in the transcription and translation into proteins. It's an amplification-free approach, no reverse transcription, no PCR amplification, which means that we study the RNA as is. It is great for research, but if you want a cost-effective tool, high throughput, you will look at cDNA sequencing. cDNA sequencing is what we call RNA sequencing with short reads. It's exactly the same thing. You will still be able to look at the fusions, at the isoforms, and quantify them precisely. The third option is to look at single-cell RNA sequencing, which is the second part of this talk. So we're gonna today focus specifically on cDNA sequencing. We have a whole workflow that starts from library preparation. You need as much as 10 nanogram of polyadenylated RNA or 500 nanogram of total RNA. We have kits to do this step. Then you will be able to sequence on our PromethION instruments. The PromethION instruments come in 3 flavors, the P2 with just 2 flow cells, the P24 with 24 flow cells, and the P48 with 48 flow cells. Each flow cell will generate up to 100 million reads, which will enable you to get enough depth to study several samples at once. Then we are going to go into the analysis, and we offer an analysis workflow called WF Transcriptome on our Epitomy platform. The files that are generated by Epitomy WF Transcriptome are compatible with third-party analysis tools if you want to refine your analysis. So first, how does the long-read cDNA sequencing compare to the short-read approach that is vastly used? We have two independent studies. One is peer-reviewed, Klinos and collaborators, the other by Chen and collaborators, all both do the same. They will compare from the same samples short-read sequencing and long-read nanopore sequencing. On these graphs, the authors have plotted the results of the transcript expression on the y-axis for the nanopore result and the x-axis for the short-read sequencing results. And you see there that we have an excellent correlation, meaning that gene expression done either with short-read or with nanopore reads will yield the same results, and you don't therefore compromise using long reads versus short reads. But on top of this, what is really interesting for researchers will be to look at the cost efficiency of using a long read approach. And here we are providing just estimates for you. It might change depending on the regions where you are. But if we look at long read and short reads, we're going to see that 10 million of long reads, which is equivalent to 50 million short reads. Because if you remember one of the first slides that we saw together, the long reads span the entirety of a transcript, while with a short read, you will need several short reads to capture the same information. So looking at 10 million long reads is equivalent to 50 million short reads. And you here have an estimation of the prices. So I come to ask you, how much do you pay for 20 million, for 100 million short reads? Think about it and look at those prices. Now, there's something else that is very attractive. And I want to go through this because this is very different from what you're doing today. If we take this PromethION 24 that holds 24 flow cells and can sequence 24 flow cells in parallel, you don't have to start all 24 flow cells at first. So let's imagine you have a small project with 5 samples for which you need 20 million reads per samples. You're going to start with 1 flow cell, and you can absolutely run only 1 flow cell. But let's imagine that a colleague of yours is coming and have 25 samples now, and they want to sequence those 25 samples at 20 million reads per samples. They can then mobilize 5 flow cells and start sequencing those 5 flow cells even 2 or 3 hours that you've started your 1 flow cell. And finally, if you have a large laboratory running several different applications, you can run all your applications at the same time on the same instruments. You can have a case where you can have 5 flow cells for RNA-seq, 25 for 20, sorry, for single cell at the same time, and there is no problem with this. So you have a lot of flexibility because each flow cell is completely independent and can be started at any point during a run, even if there's already 10 flow cells that are running. And the prices I've shown you are not dependent of filling the whole instrument. You will have access to the same price regardless of the number of flow cells that are running in parallel. Lots of publications exist using long-read sequencing, and I've already shown the utility of this in transcriptome analysis. You will see that most of the publications that we are showing are very recent, because this is really a recent trend. But authors and researchers are rediscovering or discovering the interest of those long reads. And this is an excellent example by Eberle and collaborators. If you look at this gene, TNF-SF12 in Alzheimer's disease, you can see that at the gene level, the gene expression is relatively constant. You don't really see a difference. Now, if you look at 2 isoforms, the 219 and the 203, you will realize that these isoforms are differently regulated, and they are even regulated the opposite way, whether you consider Alzheimer patient or the control patient. So it's really crucial to look at the transcriptome at the isoform level to capture, to understand it fully. And besides isoforms, you'll be able to look at fusions as well. Fusions are really important in cancer, and we know that fusions are driving cancer, but we have difficulties to know exactly their role and even their importance in the big picture of cancer. And you have one example here by Kioza and collaborators working on hepatocellular carcinoma, where they have discovered new fusions, and in particular fusions of the hepatitis B virus and its expression in this particular type of cancer. All this opens a lot of doors in understanding diseases and maybe discovering new treatments and therapeutic avenues. But let's go to the second part of this talk, and let's look at single cell coupled with long-read sequencing, or when resolution meets resolution, as I titled it. Single cell has been elected method of the year in 2018. And single cell has been pivotal in the discovery of new cell types and in understanding diseases. Here on the right-hand side, you have this example of the discovery of ionocytes, a publication from 2018 by Montero and collaborators. They've unraveled ionocytes that are a cell type that you find at a frequency of about 0.5% of the cell total in lung. This cell type is really important because it's involved in cystic fibrosis, and obviously revealing this cell type means that we have a new way to understand cystic fibrosis and find new treatment avenues. But now, most of the popular approaches to look at single cell at high scale do capture the molecules through their 3' or their 5' end, which means that when you apply short reads, as you see on this picture, you're going to sequence the 3' or the 5' of the transcriptome, of the transcripts. But you're going to missing all the information that we see here on this example, where we have a gene that is very complex with different exons and different transcripts. We cannot find this with short reads. This is where nanopore reads make a difference. You see here at the bottom that sequencing the same sample with long reads will enable to unravel the various isoforms that are present in each cell. So we're going to not only be able to reveal those rare cells, but be able to study more precisely what they're doing and their function. But let's look at an example. In this paper, Peng and collaborators apply long-read sequencing using 10x genomics to prepare their samples. And what you see there is that we have this high-scale, large number of cells that you get from a classic 10x experiment. They were able to cluster their cells, as you would also do with short reads. And they were able to look at SF3B1 gene expression. This gene encodes the largest subunit of the splicing factor 3B protein complex, and it is critical for the spliceosome assembly and mRNA splicing. So when we look at its expression, it looks homogeneous in every cell types. Now, SF3B1 can have mutations that will drive the appearance of chronic lymphocytic leukemia. And the authors, by using long reads, were able to plot those mutations. Here you have a K700E mutation that are not expressed in all the cells. They were also able to plot another mutation that results in a truncated version of the protein. And they were able then to look at what's happening in those cells as compared to the normal cells, and they were able to unravel what are the cascade of genes that are regulated by this gene, opening new avenues to find new therapeutic targets or understand the disease very simply. How did they do that? They used the 10x genomics Chromium system to partition their cells in a first step. And then, using only 10 nanogram of the cDNA after the generation of the gens, they were able to run nanopore sequencing. We have an end-to-end workflow on our website compatible with the 10x approach that will guide you through the generation of libraries from either single-cell 3' or single-cell 5', as well as Visium spatial gene expression if need be, and sequence those libraries and then finally analyze them. They run on the Promethium series of instruments that we saw earlier on. And for the analysis, we provide you with a workflow called WF single cell that will give you all the information you need, including the UMAP projection, will give you isoform count matrix, gene expression matrix, and that is completely autonomous, doesn't need any short reads for barcode assignment, and generates files that are compatible with third-party generalist ones, such as URAT, or specialized ones for which there's a lot of options, and we can talk about during the Q&A if you want more information. In collaboration with 10x Genomics, we've provided a comparison of short reads and nanopore sequencing in order to determine if we got the same results from a given sample. And you see there the results of this experiment. You see first on the top left the gene saturation curve that shows that whether you're using long reads or short reads, you're going to detect the same number of genes at a given depth. This is really reassuring because it means that both technologies perform similarly, and you will get at the very least what you get with short reads. We also see that the gene expression profile is very reliable and completely concordant between both methods. And finally, on the right-hand side, we see that the cell types and subtypes that we discover with both technologies are the same, and we retain the same cell populations. But what you cannot capture with short reads is what is described in this publication by Xiao and collaborators. They were looking at kidney cancer, and they were looking at its microenvironment. So here we see that from a patient, they were not only able to detect the normal and the tumoral cells and the progression, the tumoral progression, but they were also able to look at isoform utilization by the tumor cells, and they were able to show that the different tumor clones are using different isoforms, resulting most probably in actually a very different biology in each of the clones that will need, for instance, if it comes to therapies, combo therapies, you can probably not treat the same these different clones with the same drugs. If you look at the publication, you'll read that they've seen that it's not only for HOOK2 this difference in isoform utilization by the different clones, but it's extremely complex, and that you're going to have various genes, various isoforms in the different tumoral clones, and that right now we don't really know what is their role, but it's a fantastic tool to start to understand what's going on in this cancer. So you might think maybe it's only valid for cancer, but we also know that in the central and peripheral nervous system, there are genes that are with up to 30 different isoforms that are expressed. And right now, their role is very difficult to understand. Long reads offers you this opportunity. And here at this publication by Chen and collaborators unravel nearly 17,000 novel isoforms from the retina. Not only do they find those novel isoforms, they also found new fusions that they didn't expect really to find. And they show that this has an importance in understanding the cell role. Here they did two projections. So they did short reads and long reads at the same time from the same sample. We see that the general population structure, cell population structure, is conserved with both approach, but with long reads, they come to detect the specific subtypes of cells that they couldn't discriminate with short reads. And for this, they use the information that they got from the isoforms. So this means that if you're trying to do a cell atlas, if you don't look at isoforms, you might be missing some cell subtypes. In summary, I hope I've convinced you that using long-read sequencing and nanopore sequencing in particular, you'll be able to unravel more information from your single-cell experiment. You'll get isoforms information. You'll get fusions and SNPs information. You'll be able to do this with a single tube, a 3 hours library prep after the 10x initial isolation of cells. And you'll be able to do this in a very flexible manner on your PromethION instrument, sequencing at the same time single-cell long-read samples, as well as RNA-seq samples, as well as DNA samples. And to summarize the content of this talk, we have several approaches when it comes to RNA. So don't get too focused on what you want to do. By combining bulk cDNA-seq, maybe the direct RNA-seq, and single cell RNA-seq, you're going to be able to unravel information that you've never seen before, even in bulk, just because with the power of long reads, we have a better mapping of the information. Thank you very much, and I'll be happy to take your questions now.
Once again, thank you to our speakers for their time today, and thank you everyone for watching.